We're ready to start. I'm going to share my screen. Okay, so I'm going to give this short lecture about fluorescence and fluorophores, the different types of fluorophores, and a little introduction about fluorescence. So fluorescence is a cycle of absorption and emission of photons. Only some molecules can fluoresce. They're called fluorophore or fluorochromes. And what happens is that the molecule absorbs a photon of a certain wavelength, and then it gets excited. There's like all sorts of excitement and shaking and vibrations, and then some energy is lost, and then the molecule spits out a photon of a different color, which has less energy because of these vibrations. So in our samples, the fluorophore acts like a light source. So this is like a little lamp. Every fluorophore is like a little lamp. And because of these vibrations, we have two different lights. We have the excitation light, which is the one that is being absorbed and excites the fluorophore. And we also have the emission light, which is the one that the fluorophore spits out to go back to a more stable state. And the Stokes shift is the distance in nanometers between these two peaks. You might find sometimes the Stokes shift. People talk about, oh, I have a fluorophore with a big Stokes shift. It means the excitation and the emission curves are far away from each other. So this diagram is shown very often. It's called the Jablonski energy diagram, and it shows what happens to a fluorophore when we shine some light on it. So the fluorophore can be in different states. It could be in the relaxed state or ground state. This is what happens here. Basically, we're not shining any light on it, so it's not excited, and it's called the ground state. And this is a stable state, so it can stay like this for quite a long time without bothering. But it could also be excited, so now we're shining some light on it, and as I said, everything gets all excited and starts vibrating. And this is also called a singlet state. You might find this. This is basically the excited state. And this is unstable. So this is not something that will last for a very long time. Then we have all sorts of excitation and we lose some energy. So we go back down a little bit in a different level of energy. And there, either we fluoresce and we send some emitted light and we go back to the ground state, or we can go to yet another state called the dark state. It's also called the triplet state. And this is some sort of a parallel way. So we do not have only excited or not excited. We can also go into the dark state. And what happens in this dark state is that the fluorophore is excited. It has more energy than at the ground state, but it is stable and it does not fluoresce. Okay? So sometimes it can release some energy and fluoresce, but otherwise it can go back to the ground state without fluorescing. But while it is in the triplet state, it just stays there for a little while. So we are going to talk about these different states. Of course, the ground state and the excited state, it's easy. That's what we talk about all the time when we talk about the fluorophore is excited or is not excited. But the triplet state, the dark state, has an influence also on what we do as microscopists. And this is why I'm mentioning it here. And we will come back to this, why it matters later. So I also want to mention a few words that you may use around or you may hear around just to make sure that you know what it means. So luminescence is this word comprises all the phenomena where light is emitted. That includes fluorescence. So it's not only fluorescence, but it includes fluorescence. Chemiluminescence is luminescence that is not triggered by excitation light. So we can have a chemical that can absorb, there's a chemical reaction, and then it emits light. This is called chemiluminescence. And bioluminescence is all luminescence, so where light is emitted, but it's natural. So this is what is found is all in all nature, basically. Fireflies and corals, you have bioluminescence. So it's exactly the same thing. It can be triggered by a chemical reaction, or it can be triggered by light, but it's what you find in nature. And so phosphorescence, fluorescence are luminescence phenomena that are triggered by light. Phosphorescence, it's simply that it takes quite a lot of time because it goes into the triplet state. So it takes a lot of time to go back to the ground state and release its energy in terms of photons. So this is why when you have a phosphorescent watch, for example, you can shine some light on it, and maybe for 10 minutes later, it's still trickling down its photons. So the transition to the dark state, the fact that the fluorophore is going to either go back to like emitting and going back to the ground state, or it's going to go into this parallel life of the triplet state is, and again, triplet state and dark state is the same thing. It is a statistical process. So sometimes it will go there and sometimes not. So when we shine a very strong light for a long time on fluorophores, we increase the risk. So it's a statistical thing. So the more cycle the sample does, and the more there is a risk that some of the photons will go into the dark state. And I remind you that when they are in the dark state, they sort of get stuck there for a while. So when we image our sample with a high illumination power, we have a risk of sending the fluorophores in a dark state. And what happens then is that it looks like it's bleaching, because more of the photons of the fluorophores are excited, but they do not fluoresce. And then they stay there in this dark state, but it's not possible to re-excite them. So the sample slowly, the fluorescence goes down, but it's not because it's bleaching, it's because a higher proportion of the fluorophores go into the dark state. So when we image with high laser power, it is better to illuminate often, but more than go for a high illumination power and very often and short time, more than going for a long time. Because when you go for a long time, statistically, you will send quite a lot of your fluorophores to the dark state. So it's better to have a high illumination power for a short time than a low illumination for a long time. And this is something that is important for us to figure out, what do I do? Do I increase 10 times my laser and go very, very short, or do I do the opposite? It's better to actually have a high laser power for a short time. And it's the same thing when we use single point confocals. It's better to use frame averaging instead of a slow scanner speed, because when you shine for a long time on a fluorophore, it has a risk of, or statistically, a number of fluorophores will go into the dark state. So it will look like you bleach. So now we can have a look at photo bleaching and photo damage. What is that? When oxygen reacts with fluorophores that are in the excited state, they get this is very difficult to say, irreversible destruction of the fluorophore. So it is irreversibly destructed. There's no way to go back. Okay, so this is when we have the fluorophore in the excited state and then oxygen comes in. Additionally, on top of destroying the fluorophore, there is a production of reactive oxygen species, and these species will damage the fluorophore. So we have like two problems with shining all this light and getting the oxygen in. We destroy our fluorophore, so we bleach the sample, but on top of that, we destroy the sample as well. So we can, these oxygen species, they can destroy macromolecules. So they can, for example, or modify them. For example, you can have like mutations in DNA and things like this from these reactive oxygen species. So this is the reason why we get both photo bleaching and photo damage of the sample. So how do we prevent this from happening? We can protect our sample chemically by using oxygen scavengers. For example, the anti-fade reagents that we have in mounting media this is what they do. They prevent oxygen from destroying the fluorophore and from creating reactive oxygen species. We can also use a physical protection. For example, when we image in storm and we want to avoid that the sample is bleaching, we put a cover slip on top. And just a cover slip will prevent the oxygens from basically getting into the medium and attacking the fluorophores. So these are two different ways that we can use to prevent bleaching and photo damage to our sample. And it's important to see that bleaching is not linear. So basically, when you start bleaching a sample, it will bleach very fast to start with, and then it will slow down. And what that means is that you should not look at your fluorophores in the eyepiece. So we will teach you here a way to find your sample without looking at fluorescence in the eyepiece. And I think you will get used to it very easily because all our users get used to it. So Fazal has an advantage here because she uses it all the time. But it is there is no need to look at the fluorescence in the eyepiece. So you can look at with transmitted light and then you image it. And so you do not start with bleaching your fluorophore when you're looking around and not acquiring any images. So what type of fluorophores do we have? There are two types. We have those that are produced by chemistry, so chemical fluorophores, and we have those that are produced by the cells, and these are proteins. And then, of course, we have autofluorescence, which can be all sorts of different things. So we have like proteins, macromolecules, we can have metabolites, like, for example, vitamins, we can have pigments, etc. All these are inside the cells. So the organic dyes or chemical fluorophores can be, I'm sure you know these names. All these are chemical fluorophores, and they are developed very, very much. So there are tons of them. You can mark the DNA, you can mark the membrane, cytoplasm, mitochondria, you know, all the different compartments in the cells. You can use them in live cells or in fixed cells. So there's really a very wide variety of fluorophores that are chemicals and that you add to your cells and then they get inside and then they label. And this field is really in a very strong development. So it is actually quite useful to have a look like if you would like to, if you see, for example, that your fluor bleaches fast or it's not very good or what, just write to the company and say, do you have a better one? This is what I'm doing. Please suggest something. And they will be very happy to help you because then you're going to buy from them. So it's important to try to optimize and get the best fluorophore instead of just picking the first one that comes in. So amongst chemical fluorophore, we have some specialized ones, for example, the ratiometric dyes, and in this one, click. So this is, for example, an example of flip calcium 5, which in neurospheres in here, where you can see the calcium pulses going through, and then the fluorescence is increasing together with the calcium. So then you can quantify this. So some of them change their excitation or emission spectrum, depending on, for example, the pH or the calcium concentration or the potential across the membrane for mitochondria. So the really nice specialized chemical fluorophores are available. Other fluorophores that are a bit special are those that blink. And so what happens for these fluorophores, like the one that is in here, is that they are actually prone to going to the dark state. So some fluorophores like going to the dark state more than others, and then they stay there. And so you can see them blink. So you may see, basically, they get excited and then they go dark and then they wait there for a while, then they come back and they bling, you get a bling before they get excited again. And this is something that we can use in super resolution in Storm and Palm. And that is very useful because then it's easy to identify each fluorophore one by one, and then you get super resolution. So we'll talk about this during the super resolution lecture, but some of them are specialized in that they blink. And then the second type of fluorophores are the protein fluorophores. And so we have a plasmid with a cDNA encoding the fluorophore. And then we usually fuse it to the protein of interest to tag it. And then we transfect the cells. And so the cell itself is expressing the fluorophore with its own machinery. And And we can then follow the specific molecules in live cells, for example, or fix and image it afterwards. But unfortunately, this requires overexpression of a highly modified protein. And this is, unfortunately, we do not do enough controls to see if the modification of the protein and the fact that we are basically really overexpressing this in the cell actually have artifacts. But there is an estimation, and you can see in this paper, an estimation that about 30% of all fusion proteins actually have artifacts. And it's really important to keep a critical mind when you use a fluorescent protein. For example, never image the brightest cells, because the brightest cells are those that are very likely to be seriously not in a good shape. Okay. For example, there are a lot of issues where the maturation process can be affected if the folding the fluorophore in the ER takes longer than folding the protein, then the whole thing is going to take much longer than normal, and that may affect the function of the protein itself, okay? Or the localization might be different, or the interaction between proteins might be different. So there's a lot of possible artifacts with fusion proteins. So the proteins have also been evolved a lot, and I think I spoke a little bit about this in the exercise where you have to find the exact name of your fluorophores. There's a lot of evolvement of the proteins. It's really it keeps on going, and the field is very much going into evolving the proteins towards the red and the far red, and so there's a vast range of red and far red proteins. They all have different spectra, so you really need to check the spectra, and we get lots of different better fluorophores that come up brighter, more stable, etc. So we have also specialized fluorescent proteins, for example, photo-switchable. So they can go from one color to another color. Or we have, this is an example of MKD that goes from green to red. So it's green, and then when you shine some light, it turns to red. And so that means that in this live zebrafish, you could follow these cells. If they move, you could follow them throughout the fish because they are green, whereas the others are red. You can have also photoactivatable fluorophores proteins where you can turn them on and off. And the DRONPA, for example, is one of them where you can turn it on and off depending on the light you shine on it. And so this is KAD conversion in zebrafish from green to red. Oh, it's playing again. Then we have like little tricks that we can play with the fluorescent proteins. For example, we can encode 2 halves of the protein, and this is called bimolecular fluorescence complementation. So where each half is not fluorescent, but when this molecule A and molecule B, so these are the protein of interest, when they bind to each other, then the fluorophore is reconstructed basically, and it starts fluorescing. So we only see the fluorescence here when the protein A and B of interest bind to each other. So it's very convenient. So it shows protein-protein interaction, and it goes from non-fluorescent to fluorescent. That's called bimolecular fluorescence compensation. Another trick that we can play with fluorescent proteins is FRET, so faster resonance energy transfer. And what we have here is that we have a fluorescent protein fused to a protein of interest and another fluorophore protein fused to another protein of interest. And of course, each of these fluoresce in their normal way, normally, but when the 2 proteins of interest come next to each other, then the 2 fluorophores come close to each other, and in that case there is a transfer of energy. So they go into a vibrational state. So it's not the case that this one will excite this with its emission light. It's not like this. We're exciting here, and then there is vibration of the 2 molecules, and that one gets excited and it starts emitting. So here is an example, maybe, yes. Here an example where we have cells with a histone methylation FRET sensor. And so in these cells, when the cells are red, so you can see it's histone, so it's inside the nucleus. And when the nuclei are red, then we have a high histone methylation. And when we go to blue, that is a low histone methylation, and then in green is in between. And so you can see in here that the nuclei are going to change from blue to red when the cell divides, and then afterwards they go back into blue. So we can quantify the histone methylation in live cells using this FRET sensor. Very useful. Another example of a FRET sensor here is one molecule which fluoresces in a certain way when it's when the molecule has a certain conformation, but when the molecule is folded, for example, if it gets phosphorylated, then we get FRET happening between the two fluorophores, and then the colors change. And so this is what we have here in the focal adhesions, where we can see the tension. This is a tension sensor using FRET. So all these are tricks that we can do with fluorescent proteins. Something else that not everybody is aware of is that fluorophores have, as we have seen, an excitation spectrum, and they also have an emission spectrum, but they also have a lifetime, which is measured in nanoseconds. So what is the lifetime of a fluorophore? It's basically if you're shining a pulse, a very short pulse of excitation light on the fluorophore, it will get excited, or on the sample. Some of the fluorophores will get excited, and then they will start emitting, right? But then they slowly stop emitting because you just send a very short pulse of excitation. So they will start emitting, and then slowly they will go down. And this curve of the emission, that intensity that goes down, is very typical of the fluorophore. So different fluorophores will have a different curve depending on the fluorophore. And so this is some sort of a signature for each fluorophore, as much as the excitation spectrum and the emission spectrum are a signature of the fluorophore. On top of that, when the fluorophore is in a different environment, this curve changes. So even if you have one fluorophore and you change the pH, for example, the curve is going to change, which means that you can actually measure the environment of the fluorophore just by looking at the lifetime. And so this is also very useful. And in this example, we can see, so I don't remember, I think I had it, no, maybe I forgot to write what it was. I forgot what the fluorophore is, but I think this is a calcium sensor. And what we can see is that when we send the pulse to these cells, then they suddenly get a completely different color. It's actually not a color. This is color-coded. This is the change of lifetime of this fluorophore. And then slowly it will go back to going back to blue. Okay, so it will go back to its original lifetime. Then we have techniques where we can combine these two techniques, the chemical and the fluorescent protein. The problem with the fluorescent protein is that it's very disruptive for the protein. So the fluorophores take a lot of space. Some of the proteins are small, and so the fluorophore like doubles up the size or something like this. It's a hindrance for other molecules to bind and all that stuff. It leads to artifacts. And for that reason, what you can do is use click chemistry instead of SnapTag. All these things have the same genetic code expansion. All these things have the same principle, which is you are overexpressing a protein with just a small modification. For example, it can be just one amino acid that is different. So you're going to modify slightly your protein of interest with a very, very small modification. So it's super tiny, it doesn't take any space. But this modification will allow you to put a chemical on the cells, and this chemical is going to bind to this modification. So now we're going to basically label proteins inside the cell with a chemical, and the chemical is going to bind to that protein specifically that you have overexpressed. And then it starts fluorescing because the chemical itself is fluorescent. So this is extremely specific, because you only label the protein that you overexpress, and it has a low disruption of the protein conformation, so it's very good, but it anyway requires overexpression of the protein. So click chemistry, SnapTags are very useful, and you can do this in live cells as well. So one thing that I want to say is that as always, you need to be critical with what you inherit from your lab. It's unfortunately the case that we have before us, when we come to a lab, generations of researchers who were maybe, you know, not fully aware of what is possible. Each of us, we try to do our little soup there in our corner, and sometimes we get bad ideas. And so you also inherit the bad ideas. So you need to also be critical for what you're getting. If someone tells you, just use that one, you know, you can go and check, is there a better fluorophore than this? Maybe this one bleaches very fast. Especially if you notice that there is a problem, you should definitely go and look for something better, and you will do your lab a favor by improving their protocol. So this is a website that is super useful, fpbase.org. So I think some of you have already used it. So basically, you can use the interactive chart. You can go to the in the advanced search. So the advanced search and the interactive chart are very useful. But in the menu, you will find that. And what you can do is say, okay, I would like to find a fluorophore of whatever excitation wavelength I would like and whatever emission. So you just select the range here to filter. And then you will see this chart change and show you only the stuff that you're interested in. And then you will get all these little dots here, and you can click on each of them to find the original paper that is linked, and also the sequence of the protein. So this is really, really useful. And you will find also the excitation and emission spectra, and you can, using this, you can find better fluorophores that match, for example, your microscope. So you will also find quite often the fluorescence lifetime, and here you see that you have more filters that you can filter on. And for example, I spoke about the maturation. How long does this fluorescent protein take to fold in the ER? If you're studying very fast processes where there's a very fast transition and turnover of the protein, you want to be careful with the maturation. Sometimes you have to be really careful with the stability, for example, the photostability or the pKa, or the brightness. So you can find the brightest proteins. So this is the maturation time. So how long does it take to fold? And also the photostability, so how easy it is to bleach it. And the excitation, the EC is excitation coefficient, which means how easily it absorbs light. Clearly, you would like it to absorb all the photons that you shine on it so that it's efficient. And the quantum yield is, for each photon that is absorbed, how many photons does it emit? So quantum yield of 1 would be perfect. So this is the quantum yield. And the brightness is the multiplication of the two. So I like to look at the brightness so that I figure out, okay, what is a green fluorophore excited by this wavelength with this emission that would be the brightest? Then I can find it that way. Another thing that is important to consider is the oligomerization. So there is a little click box that you can click to say, show me only monomers. Even weak dimerization is likely to disrupt the target protein function. So this has been a problem with old fluorophore proteins where they were binding to each other, basically. So you get the two fluorophores binding and the protein of interest is dragged with. So this is not good. And this is the reason why we want to only use fluorescent proteins that are true monomers. So you can look at the fpbase.org and look at the interactive chart, and I'm sure you will discover new proteins that you didn't think were much better than your own. So it's the same thing. You should pick also the best chemical dye. Unfortunately, we do not have the same tool as the fpbase.org to choose or to compare the chemical dyes. So we have exactly the same thing that we can consider: photostability, the brightness, etc. But of course, the maturation time and the oligomerization is only for proteins. But otherwise, the same applies to everything else. What you can do to find them is to go to the molecular probe handbook. There's a lot of fluorophores that you can find in here, lots of questions, and you can ask the company, Thermo Fisher, for help in finding the best fluorophore for your own sample to replace, for example, the one that your lab has been using and maybe is not the best possible now. So another part of fluorescence is, of course, autofluorescence, and this comes from many different things. But it's sometimes an annoyance and sometimes it can be useful. So we have to see it in a positive way. So in this sample here, we have treated with Sudan black. And you can see that the sample is clearly improved. Sudan black is very good if you have some paraffin embedded samples. So there is a green autofluorescence that comes up and that can block it. So this would be sort of a chemical treatment. So there are different chemical treatments to get rid of autofluorescence, but you can also use spectral unmixing. So we will use a little bit, talk a little bit about this later. And bleaching can be a way also to get rid of autofluorescence. And yes, autofluorescence can also be an advantage where you can image the sample without labeling it. So we'll talk about all this during the sample preparation module with Gabi. So now I want to conclude this little lecture. So please choose bright, photostable, and monomeric fluorescent proteins in the FP base. Go and compare the fluorescent proteins that you're using with what is available, and maybe you will get some nice surprises that it's easy to get 2-3 times more fluorescence by just changing the fluorophore. Be critical for what you get from your lab. And it's sometimes has been chosen randomly. Sometimes it has not, but sometimes it has. So I think you don't have to during all your PhD a fluorophore that is not efficient. You can also do the job and try to figure out if there is something better. So always check for the if you have efficient excitation and emission detection on the microscope that you use, and also make sure that you pick the fluorophores to have no bleed through on the microscope you use. And you can ask the companies, don't hesitate to write the companies to say, hey, this is what I'm using, this is my experiment, do you have anything better? And do you not just pick a fluorescent from the fridge just because it's there? This is doing yourself a disfavor. Okay, I think I am now finished, and I would like to know if there are any questions. I think I have taken a little bit longer than I had planned. Are there any questions from you guys about the fluorophores? Yes, Sadaf.
Thank you. I would like to ask about the method you mentioned, for example, click chemistry or snap tag. So is it possible to do that for RNA as well?
Well, you know, in the snapback and click chemistry, you have to modify the protein of interest and overexpress it. So there are some techniques now where you can have, for example, maybe this is what you mean, and I'm not sure, but basically, if you have an RNA, you can put a complementary molecule, like a complementary RNA that you have designed yourself, and then you click a fluorophore on that. That exists already. It's not through the same thing as modifying the protein and then clicking on the modified protein, but it's the same principle.
All right, and just to clarify this for myself, so in FRET, you mentioned that there are two molecules binding to two fluorescence proteins, and then when they become when they get closer to each other, those fluorophores, they shake and change color as a result
They do not change color. They do not change color. But what happens is that if you have two fluorophores, so you have a certain excitation and emission spectrum for each of them, right? And they are different. And so when you excite fluorophore A, it emits, right? But the fluorophore B is not going to emit, for example. But now when the two proteins get together and the two fluorophores get together, you're going to excite fluorophore A, and the fluorophore B is very close to it. So when we are in this excited state where the molecules are shaking, there's a lot of vibrations there, and these can induce the fluorophore B to go into the excited state, where it is not unstable and start fluorescing. So the result of that is that you're going to excite A, but B is going to be fluorescing. It's not that the fluorophores are changing color. It's just the consequence that you're exciting A and then B fluoresces. So if A transfers its energy to B, A is not going to fluoresce because it loses its energy by transferring it. And so what happens is that in essence, if they are far away, you're going to excite A and you will get the emission of A. But when they get close to each other, you continue exciting A, and the emission of A goes down a lot, and B starts emitting.
Okay. Yes, makes sense.
The light that you're getting is different, but it's not because the fluorophores have changed color.
Because they're close together. Okay.
Because they're close to each other and they get co-excited.
Thank you.
There. Other questions? Someone has a question here. I can. Yes. Shu, did you have a question?
Hi, Sylvie. Can you hear
Hi.
me?
Yes.
Yes. So I have one question. So it happened to me when I use when I ionize RNA molecules, I use DAPI to to localize a nucleus to help me to assign the RNA molecules. And in the meantime, I also tried with a membrane dye to ionize the RNA.